Hi, my name is Andrew Goldstein. I'm a professor at UCLA, and I'm really excited to share this new work with you. We've been working on it for more than 6 years, a lot of really cool discoveries and trying to figure out how things piece together. And eventually we were able to come up with what I think is a really fun discovery, and I'm excited to be able to share it with all of you now. This paper was led by two outstanding graduate students from my lab, Jenna Jaffaloni and Preston Kroll, with a lot of support from collaborators at UCLA and really all around the world. The normal prostate epithelium is made up primarily of two layers, basal cells and luminal cells. During cancer initiation, there's an expansion of malignant luminal-like cells that express the androgen receptor. But after AR-targeted therapies, some resistant tumors emerge with a loss of luminal differentiation, and this process is often referred to as lineage infidelity or lineage plasticity. So luminal differentiation occurs in development in response to tissue repair or inflammation or cancer initiation from basal cells. And the loss of luminal differentiation can occur during disease progression, in response to AR-targeted therapies, or as a result of gaining specific mutations. Two really important questions to ask are, does the differentiation state matter for prostate cancer? And what actually regulates prostate differentiation? Results from many studies show that lineage phenotype matters. Here, as an example, a set of metastatic castration-resistant prostate cancers were classified into basal or luminal subtypes based on gene expression. The basal-like tumors have low AR signaling and, importantly, a really poor clinical response to AR-targeted therapies. So the differentiation state in prostate cancer matters because it's associated with response to therapy and patient outcome. Specifically, the more luminal-like tumors are more responsive to androgen receptor blockade. Therefore, it's critical to understand what makes a prostate cell more or less luminal. Now, prior studies point to epigenetic regulation of prostate lineage identity. Epigenetics is, in many ways, governed by upstream metabolism. And so we wanted to understand if upstream metabolism might actually contribute to prostate luminal differentiation. We performed metabolic profiling and nutrient tracing in distinct prostate cell types. We found evidence of lineage-specific metabolic features in the prostate, including finding terms like pyruvate metabolism and oxidative phosphorylation enriched in the luminal cells. To summarize a lot of data that I'm not going to show here, we found that distinct cell types have distinct metabolic preferences in the prostate. And so if the cell type dictates metabolism, does metabolism change during luminal differentiation? Using a model of basal to luminal differentiation, where the luminal marker K8 goes up and the basal marker P63 goes down, we asked how labeled glucose carbons get incorporated into glycolytic metabolites, TCA cycle metabolites, or nucleotide intermediates as cells become more luminal. We found clear evidence that as luminal differentiation increases, we see increased labeling from glucose into TCA cycle metabolites, indicative of pyruvate oxidation. So pyruvate oxidation goes up as cells become more luminal. Now, is this just happening, or is it functionally contributing to luminal differentiation? Pyruvate oxidation is driven by a complex called the mitochondrial pyruvate carrier, or MPC, which can be blocked pharmacologically with the small molecule UK5099 or through genetic approaches to delete MPC1 or MPC2. We found that MPC inhibition or knockout led to reduced expression of luminal markers like K8 and increased expression of basal markers like P63. And this was done in a wide range of organoid, PDX, and cell line models. So blockade of pyruvate oxidation impairs luminal differentiation. But we wanted to understand the mechanism. How does pyruvate oxidation influence luminal differentiation? When pyruvate is blocked from entering the mitochondria, more becomes available to get converted into lactate. And we found more lactate in cells after MPC inhibition. Simply supplementing cells with lactate was sufficient to reduce K8 and increase P63. Using nutrient tracing, we found that lactate supplementation and MPC inhibition actually had very different effects on metabolism, even though they both disrupt luminal differentiation. So we wondered if the phenotype was driven less by metabolism and more perhaps by changes at the epigenetic level. Now, interestingly, studies have suggested that lactate can act as an inhibitor of histone deacetylase activity. And so we used ATAC sequencing, finding in one example that MPC inhibition or lactate supplementation increases chromatin accessibility at the promoter of the basal lineage gene P63, and actually in the vast majority of basal lineage genes. So intracellular lactate accumulation regulates lineage-specific gene expression through changes in chromatin accessibility. And since lactate influences luminal differentiation, we wanted to know if it also influences response to AR inhibition. In studies where clinical tumors were biopsied and sequenced, and then patients were subjected to hormone therapy and classified as exceptional responders or non-responders to AR-targeted therapies, we found that MPC expression was much lower in the non-responders. And this was actually true in distinct stages of the disease, using data from multiple different clinical studies. Using a PDX model, we blocked pyruvate oxidation or supplemented with lactate and then subjected cells to AR inhibition. While the control cells were quite sensitive to enzalutamide, modulating lactate metabolism promoted resistance to AR inhibition. So in clinical tumors, good responders to AR inhibition are more luminal and have higher levels of MPC. Poor responders to hormone therapy are less luminal and have low MPC expression, and experimentally increasing lactate was sufficient to promote resistance to enzalutamide. So to summarize, basal and luminal cells have lineage-rooted metabolic features. Pyruvate oxidation increases with luminal differentiation, but it's not just associated with differentiation, it's actually functionally contributing. If we experimentally reduce pyruvate oxidation or increase intracellular lactate abundance, we can actually drive a block of luminal differentiation. We can alter chromatin accessibility at lineage-specific genes, and we can promote resistance to AR inhibition. And zooming out and thinking about what this really means, it says that in the prostate, metabolism can modulate response to therapy. So we really need to consider what are the consequences of our treatments and what are the effects of systemic or local signals on metabolism. I want to finish with this great analogy from an accompanying News and Views by Martin Bacht and Misha Beltran. We care about the clinical response to treatment. And we know that lineage can influence the response. Now, underneath the surface is epigenetics, but even less understood, and we think just as important, is the metabolism. Again, I want to thank the authors, led by two really brilliant, hardworking graduate students, Jenna Giaffaloni and Preston Kroll, with contributions from so many different scientists with different areas of expertise. We're really grateful for everyone's contributions, as well as funding support to be able to pursue this work over many years. And I'm really proud of what we've put together, and I hope you enjoy it. Thank you.